All right. I'm excited to be here. Thank you so much for the opportunity to speak. I'm Brad Ringeisen, the Deputy Director of the newest office at DARPA. I know you've heard DARPA's name mentioned a couple times in the talks earlier today. The Biological Technologies Office was formed about 3 years ago. And so I'm really excited to sort of share a little bit about how I got to DARPA as a scientist, and then sort of some of the things that are emerging out of our office, the BTO office. So, what I'm going to talk to you today about is cutting edge research. I think a lot of you, and part of what the STIX conference today really is about, is helping people understand that the Department of Defense actually does stimulate and innovate some of the most amazing technologies that you can imagine in the country today. And DARPA is a big part of that. But part of the problem sometimes with innovative technology is sort of the hype curve, sort of the unreasonable expectations that are placed on scientists as they start to develop new ideas. So I'm going to give you a couple of stories of that are very personally affected by myself. You can see back in 2001, I had arrived at the Naval Research Laboratory where I started my career in 2000. I got a phone call from a reporter in 2001. Young, naive Brad picked up the phone. Hey, yeah, wow, that's great. I really want to talk to you. How are things going? So I had published a paper, my first paper as a postdoc at NRL, where I'd actually deposited one of the first living cells using a 3D printing technology. Okay, this was back in 2000, 2001. So Catherine Zandonella called me up and said, hey, Brad, talk to me about some of your technology. So we had deposited some bacteria. We were just starting to work with mammalian cells. I told her about some of the research that we were doing. Then I got sent a copy of an article whose title was New Body Parts at the Stroke of a Laser Pen. Okay? So I can guarantee you that those words did not come out of my mouth. But that's something, as an example of sort of the expectations that are placed on new emerging technologies that you find. So I'll show you a little bit more in detail about what I'm talking about. So like I said, some of the first living cells were being printed back in the late 1990s into the early 2000s. I can give you an example of that. This is the laser technology that's patented at the Naval Research Laboratory. And this was truly one of the very, very first experiments to demonstrate. The green cells here that you're seeing are viable living cells. You can see orange cells sort of underneath them. Those were cells that died because they actually sort of were like bugs on a windshield that sort of hit the glass slide underneath a hydrogel. So they sort of ballistically penetrated through that gel, splatted up against the glass slide. So what we realized very early on was that you sort of had to have a catcher's mitt. You had to have something soft sort of to catch these very fragile cells. If you had a soft surface, you essentially could create any type of cell pattern that you want using this cell printing technology. So fast forward 7, 8 years, we co-author a book, some review issues, and literally 16 or 17 years later, there I am shaking the hand of the ex-Secretary of Defense, Ash Carter. This was literally 16 years after some of the first experiments that I had done. So you're talking about almost 2 decades of development. Now, can we create a body part at the stroke of a laser pen? No. 16, 17 years later, we are still nowhere close to actually being able to do that. And so it's one of the reasons I decided to move on to DARPA was to maybe try to start to initiate and accelerate the pace of research. All right, case study number 2. In about 2005, I switched gears and started doing research with electrophilic bacteria. These are bacteria that can take carbon sources and externalize electrons to perform an extracellular electron transfer. So if you put those bacteria in contact with an electrode in a fuel cell, you can actually generate electricity. Okay? And so here's some nice pretty pictures of what some of those bacteria look like. The first paper I published in this, I actually set a power density record in the world for how much power you could get out of a microbial fuel cell. It's on the order of about 500 microwatts for a cubic centimeter, I think. So you're talking about not very much power for a small amount of volume. Very quickly, people piled onto this research and started publishing about purported bacterial nanowires that could carry electrons away from the body of the bacteria. I, as a physical chemist, was very skeptical that these could literally be wires that could actually be metallic-like character. So we entered into a bunch of experiments. We tried to measure how much current a single bacterium could measure, and you see an example of a very small microelectrode with just a handful of bacteria sort of resting on that electrode. And ultimately, there was a lot of controversy. You've probably heard of things like robot wars. Well, back in these days, there were bacteria wars where somebody thought their bacteria was best, another team thought their bacteria was best. So Physics Today sort of picked up on this and wrote some articles on this. This is what scientists sometimes do in their spare time. But what I wanted to show you was, again, literally 8 years after I started working on this research, we finally were able to characterize exactly how much current could be generated by a single bacteria. And that was, drum roll, about 200 femtoamps of current. Okay? So let's just extrapolate that out. You're talking about 1 trillion cells, 10 to the 12. Maybe you can get 0.2 amps if there's no loss in that scaling factor. Okay? So again, I'm using this as an example of sort of the pace of scientific discovery. The public often thinks you get a result, and then the next day you're going to be able to apply it into a new product that's going to change the world. Usually, it takes a much longer time frame than that. And I think some of the presentations you saw earlier are good examples of that as well. All right, so that was my career at NRL. I literally spent 16 or 17 years at NRL, and you can see from those experiments that had lots of success, but also some sort of inherent frustrations at sort of the pace of scientific discovery. Enter DARPA. DARPA is this unique, wonderful entity that we have in the Department of Defense that is known for accelerating the pace of discovery. Things like the internet, Siri, self-driving cars. The bar is set extremely high at DARPA for success. So let me tell you a little bit about the Biological Technologies Office and some of the things that are emerging that hopefully will rival some of those previous discoveries. So I put sort of an array of some very interesting programs. Our website has much more details on all of these. I'm going to give you a very brief introduction to some of these. There's a program called Electrics that is using electrical and ultrasound stimulation of the peripheral nervous system to actually induce an immune response, to be able to actually maybe help stimulate your body to be able to fight infection or an autoimmune disease. So can you imagine one day maybe going to the pharmacy and picking up a little battery pack or a little portable ultrasound and having that rather than a pill that you would take to be able to maybe help you fight off a disease that you have? There's also a program that I'm managing called Pathogen Predators. Everyone's heard of antibiotic-resistant or antimicrobial resistant organisms. This is a tremendously large, growing threat to our society. Turns out the people at DARPA discovered that there were bacteria that had been characterized that actually go around and prey on other bacteria. And so I think something on the order of 150 antimicrobial resistant organisms can be preyed upon by these specific types of pathogens. I'm sorry, predators that prey on those pathogen organisms. There's a program called ELM, Engineered Living Materials. Might be able to revolutionize the building materials that we use to try to impart characteristics of living materials. The example that the program manager often uses is wood. Wood has amazing characteristics. It's been used for millennia as a building material, but it also has pretty significant limitations. It degrades over time. It's not very strong. So you can imagine maybe using living organisms to be able to impart things like regenerative capabilities or maybe adaptive capabilities to building materials. Now, some of our favorite programs down here, Revolutionary in Prosthetics and Restoring Active Memory. Revolutionary in Prosthetics is truly a revolutionary program. You see President Obama there interacting with one of our subjects in this program. These are people that are paralyzed, quadriplegic, paralyzed patient that has a direct cortical interface into his brain. And just by thinking about moving his arm, he's able to move this prosthetic arm. He actually gave President Obama a little fist bump. It's a very famous picture that you can see. Okay? Just an absolutely amazing technology. If you see this technology in person, you will literally leave a changed person. Restoring active memory. Raise your hand if you have a little memory problem occasionally. I'm terrible, terrible with names myself. So imagine being able to map the process of memory as you are trying to remember a process. There's some anecdotal experiments that are performed in this where they show images, 10, 15, 20 images, and they're able to record what the neural process is during that memory occurrence. And then as the patient tries to recall those memories, if you get an electrical stimulation, you actually enhance your ability and aid your ability to be able to recall those memories. And so it really is a potentially revolutionary program itself. And then finally, we have a program called Insect Allies. Traditionally, genetically modified crops and organisms take years, even decades sometimes to create. You have to go through a breeding process, crossing, through an entire regulatory process. Insect Allies is going to try to attempt to change the phenotype and the traits of these plants in one single growing season. So if there's a drought that occurs, or if there's an act of agroterrorism, we could unleash insects onto these crops to be able to impart and deliver new genes and new traits to those plants to be able to fight that threat to those crops. All right. I will dig very quickly. I didn't realize that there was a build here. I apologize for that. All right, very quickly go through three programs in more detail. This is a program because I helped develop some of the bioprinting technologies in the country. This is a program called Microphysiological Systems, or Organs on a Chip. We have performers at MIT and the Wyss Institute at Harvard that have literally taken the human body and mimicked that and replicated it on a chip platform. This will accelerate drug discovery. It will make the development process fail faster. Over 90% of drugs fail throughout the pipeline because of efficacy or toxicity. This is a program that helps to change the way that drugs are developed and screened. And I know I'm running short on time, so I'm gonna keep going. Another one of the programs in our neuroscience portfolio is the Targeted Neuroplasticity Training, or TNT program. This is a program that I just recently have taken over as the program manager. The vision here is to actually enhance cognitive skill learning by trying to sort of non-invasively stimulate the peripheral nerves. So you have the vagus nerve right here in your body. You can actually now stimulate that nerve and actually help enhance the time that it takes to become proficient at a language or a specific skill that you're trying to learn. So you can imagine for our soldiers that are trying to learn foreign languages, right now they go to very intense school for months on end. If we could help them get more proficient at those skills very quickly, that's going to help them perform their missions out in the real world. All right, and then finally, one of our newest programs is called Safe Genes. We are currently undergoing a revolution in molecular biology right now. I'm sure all of you have heard of the technologies CRISPR-Cas. These are essentially technologies that can go to a very specific location in the genome, snip the genome open, and then you can insert a very specific target gene and functionality into that organism. You can do this for yeast, you can do this for E. coli, you can do it for plants, you can do it for mammalian cells now. Okay? So this is a tremendously powerful technology that has just emerged over the last few years. Well, we have a program manager, Ringeisen, in our office that recognized the power of these technologies, but was also a little bit scared about the technologies. There are off-target effects of some of these. You can imagine if that gene editor comes into the genome and goes to the wrong part of the genome and maybe snips and edits the wrong component. Well, she's developed a program to sort of put safety bumpers on these technologies. And in three sort of categories, she's going to better control gene editing by creating new gene editors that sort of have better control, less off-target effects, and maybe some new capabilities. She's also going to develop countermeasures and prophylaxis that could halt gene editors. Right now, there's not really ways to be able to stop this process. So if it escaped into the environment, it could sort of keep doing the editing without any types of controls. Obviously not a great thing. So Renee's program, Safe Genes, is looking at inhibiting sort of unwanted gene activity. You can also imagine our adversaries in terms of the Department of Defense need. We would like to have that inhibitory process. And then finally, wouldn't it be great if we could actually remediate and reverse those genetic engineering processes? So if we were attacked, perhaps, we could actually reverse and remediate those processes to sort of make it a safer world in this brave new world of gene editing and synthetic biology. Okay, so with that, I really thank you for the time and the opportunity, and hopefully I invigorated and excited people about the types of technologies that the Department of Defense can offer. All right.